This covers chapter 25, The Origin of Life, a very brief version of that chapter. I definitely encourage you to read the condensed notes for this and skim through the chapter as well. So the origin of life probably is sparked by just the right combination of physical events as well as chemical processes. I really like this picture here, which has been seen and reproduced many times. It's an artist's rendition of early Earth and what it was most likely like. In the foreground, you see stromatolites, which are some of the oldest organisms on the planet. A lot of these kind of hot water type of environment with gases, thermal gases being released, and extravolcanic activity releasing heat and gases into the atmosphere. As Earth was cooling and condensing, this is probably a pretty good simulation of what it would have looked like at that time. So when we look at the evolutionary tree of life, it can be very well documented with evidence. The origin of life on Earth is another story. How did it begin? But before we get to that, let's take a look at some of the timeline of what happened on Earth. So approximately 4.5 or 4.6 billion years ago, or 4,500 million years ago, the Earth formed, along with the rest of the solar system. Somewhere around 3,800 years ago, the molten hot surface begins to cool on Earth. And as it cools, it condenses into something solid. Then, as we move forward in time, during the Precambrian period, we have some of the oldest definite fossils of prokaryotes, around 2.5 billion years ago, and some even date older than that. Around 2,000 years ago, we see the appearance of oxygen in the atmosphere, most likely due to the photosynthetic activity of some of those prokaryotes. 1.5 billion years ago is when we see the oldest eukaryote fossils. 1,000 million or 1 billion years ago, the first multicellular organisms, and then finally, about 700, 600 million years ago, the appearance of land and animal plants. Land plants and animals on land. Easy for me to say. And then during the Paleozoic, the colonization of land by animals. So here is the tree of life, if you will. Try to back that up. There we go. So common ancestor giving rise to bacteria, and remember archaeobacteria, two different types of bacteria, and a common ancestor giving rise to archaeobacteria and protists. Then common ancestor giving rise to a variety of protists, which we don't spend enough time on, but they are animal-like, plant-like, and fungi-like. Plants branching off of protists, and then over here fungi and animals branching off as well. So the origin of life is a hypothesis, and there are three competing hypotheses to explain how life originated. Special creation is a hypothesis. Was life created by a supernatural or divine force? Perhaps. However, in the scientific realm, this is not a testable hypothesis, and this is why it doesn't get any attention from the scientific community. If it can be tested, then it would be addressed. Remember, hypotheses must be observable and testable. Extraterrestrial origin. Was the original source of organic or carbon materials like comets and meteorites striking early Earth? Perhaps, and this is testable. What about spontaneous abiotic origin? Did life evolve spontaneously from inorganic molecules? Perhaps, and this is also testable. So let's consider what early Earth was most likely like. It's quite probable that there was a reducing atmosphere in the Earth, and it contained these gases: water vapor, carbon dioxide, nitrogen, various nitrogen oxides, so you can put in here different numbers past the oxygen, hydrogen gas, ammonia, methane, and hydrogen sulfide, or sulfuric acid. Plenty of available hydrogen and the electron that goes along with it, therefore making it reducing, and no free oxygen. So what was the energy source? Lightning, UV radiation, and of course, volcanic activity. So with low oxygen, organic molecules don't break down quite as quickly. So what's missing from the atmosphere? The oxygen part, right? It's unclear whether young Earth's atmosphere actually contained enough methane or ammonia to be reducing. Evidence does suggest that early Earth's atmosphere was made up primarily of nitrogen and carbon dioxide and wasn't really reducing or oxidizing. Remember, oxidizing is electron removing. The Miller-Urey type experiment that we're going to look at using such atmospheres have not produced organic molecules. Still, it's likely that small pockets of early Earth, perhaps near volcanic openings, etc., were reducing. So instead of forming in the atmosphere, the first organic compounds on Earth may have actually been synthesized near submerged volcanoes and deep sea vents. These are weak points in the Earth's crust, remember, where hot water and minerals gush into the ocean. So even if the entire atmosphere wasn't reducing, it's very, very likely that there were pockets of reducing habitats or environments, if you will, where this could have occurred. This is a very highly used picture to kind of graphically represent the Miller and Urey experiment. First, let's take a look at a couple of important ideas. The first idea, first abiotic synthesis, which was the last of the three hypotheses, was actually first kind of proposed by O'Perrin and Haldane. In 1920, they proposed that the Earth's atmosphere had a reducing effect. So with that, the Miller-Urey experiment was tested in 1953. And so what they had here was an apparatus, I'll show you a better picture on the next slide, where they have in a closed system a mixture of, well, let's start back down here. It doesn't really matter, it's a cycle. You've got this kind of heated water element that's supposed to represent the ocean. And then a mixture of gases, which partially would have some gases coming from the water, would be up in the tube and then coming down into this glassware, where they would have electrodes into this bubble, this enlargement of the glassware that would simulate lightning to produce energy. Within that are produced, within this mixture of primitive gases that were likely in the atmosphere, like ammonia, hydrogen, and methane. New combinations of these elements would come about and come down into this condenser where there's water, and basically it would be capturing these molecules and cooling them to the point where they could condense, and then they would capture those actual molecules into a liquid. In the liquid, when they would harvest them, they would find organic molecules. So in other words, these molecules would be formed in kind of a warmer environment, but then when cooled would condense into something that could be more stable than it would in a gaseous form. Now, what's really, really remarkable about this experiment is that organic compounds such as amino acids, of course the building block of proteins, and adenine, remember that's one of your DNA bases, and RNA bases, were formed from inorganic gases, and that's very, very remarkable. And this happens all by itself. Once they put everything in place and let it run, these things occur spontaneously. Here you see the real apparatus, and here's Stanley Miller in the background, the University of Chicago, in his younger days. So why was this experiment important? Because it produced some very important organic molecules that we see in living things from inorganic substances: amino acids, hydrocarbons, nitrogen bases, and other organic molecules that we find in living things. So again, the take-home message is from inorganic substances, organic substances were formed. So what were the key events in the origin of life? You needed to have the origin of cells, or protobionts. One idea is that lipid bubbles basically have a separating point from the inside from the outside, and it allows a space for metabolism and reproduction to occur. In regular biology, we talk about the coacervates as being a possible step in this direction. You needed an origin of genetics. Now, RNA is likely actually the first genetic material, not DNA. Why? Because it's got so many functions. It can encode information. It's in fact self-replicating. It acts as an enzyme. It's a regulatory molecule, a transport molecule. Remember tRNA and mRNA? So RNA itself would actually make inheritance possible, therefore making natural selection and evolution possible. If that doesn't make sense to you, pause and think for a minute. Why, if we have RNA, the basis of genetics, why would that make these things possible? And then, since most of the life that we're familiar with is really a eukaryote, the origin of eukaryotes would be one of the other major steps in the origin of life, and through that, the process of endosymbiosis. So remember that life is defined partly by two properties: accurate replication and metabolism. Neither property can exist without the other. Self-replicating molecules and a metabolism-like source of the building blocks must have appeared together. How did that happen? The necessary conditions for life may have been met by protobionts, which are just aggregates of abiotically produced molecules that are surrounded by a membrane or membrane-like structure. Protobionts exhibit some of these properties associated with life, including simple reproduction and metabolism, as well as the maintenance of an internal chemical environment that's different from that of its surroundings, so basically a little bit of homeostasis. Lab experiments have demonstrated that protobionts could have formed spontaneously from abiotically produced organic compounds. For example, small membrane-bound droplets called liposomes can form when lipids or other organic molecules are added to water. So again, we see some production of materials that we see in living things, almost a membrane here, from inorganic substances spontaneously. Let's take a little walk through the timeline of some key evolutionary history of life on Earth. So this is like a little clock. Here's the origin of Earth, billions of years ago, and swinging on around. So we've got prokaryotes, then oxygen-generating photosynthesis, single-celled eukaryotes, of course, are still in existence, multicellular eukaryotes, diversification of animals, land plants, and then humans. And why do we even bother putting them on there? Because we're egotistical. We like to know about our own evolution. So between 3.5 and 4 billion years ago, somewhere in here, life originated. At about 2.7 billion years ago, there was enough free oxygen in the atmosphere produced by photosynthetic bacteria that other organisms that require oxygen evolved. And at around 2 billion years ago, we see our first eukaryotes. So what about those first eukaryotes? Remember, to be a eukaryote, what does that mean? You've got a nucleus, right? You've got your DNA enclosed in a nucleus, and you have membranes or internal structures that compartmentalize those different structures inside your cell. So how did the development of those internal membranes occur? Here's your prokaryotic cell with its DNA, its cell wall, and then just inside of that is your plasma membrane. Beginning with just an infolding of the plasma membrane, just folds in on itself, giving rise to a prokaryotic ancestor of eukaryotic cells. More infoldings, DNA still in the same place. So what we have now is, as you can see, the evolution of the endoplasmic reticulum, which is kind of like the guts of the cell, or the intestines of the cell, where certain proteins are being shuttled around, and of course there are different functions of the rough and smooth endoplasmic reticulum. And then notice that this membrane expands around now a nucleus. So now the DNA is protected or housed within the nucleus. So during the development of the membranes here, we are creating an internal microenvironment, and the advantage, specialization or increasing efficiency, which can allow for natural selection because they may be better at doing their jobs than other cells that don't have these internal membranes. And this all occurred somewhere around 2.2 billion years ago, rather, not 2.2, but 2. So let's take a look at the first endosymbiosis. There were two, and the further evolution of eukaryotes. So here we're looking for our single-celled eukaryotes, the origin of mitochondria. Here's your ancestral eukaryotic cell, which then engulfed an anaerobic bacterium, but instead of digesting it, did not. And in this, there was a mutually beneficial relationship. The aerobic bacterium has a safe place to live and getting nutrients from the host cell, And the host cell is getting energy from the aerobic bacterium, which then gives us our mitochondrion. Notice the double membrane now. It had its own to begin with. And then during endosymbiosis or endocytosis, we get that second membrane. So here's our eukaryotic cell with a mitochondrion. And so if this cell can produce more energy because of the fact that it has a mitochondrion than competitive cells, then it would basically do better than those, therefore natural selection. Second symbiosis, further evolution of eukaryotes. What about the origin of chloroplasts? Same idea as we saw with the mitochondrion, but now instead a photosynthetic bacterium gets engulfed but not digested. Same principle where there is a mutually beneficial relationship, and because this guy can make energy in two ways, natural selection again. It is better suited in its environment than its competitors, therefore is going to be more successful, make more offspring. The offspring will also have the chloroplast and the mitochondria, and they will be more successful, and so forth and so on. So you end up with the eukaryotic cell with both chloroplasts and mitochondria. So who developed the theory of endosymbiosis? Len Margulis. And here's the evidence. Structural. Mitochondria and chloroplasts do resemble bacterial structure, and we definitely spent a lot of time with this a while ago. Genetic. Remember that mitochondria and chloroplasts have their own circular DNA like bacteria? And last, functional evidence. Mitochondria and chloroplasts move freely within the cell. They're not bound to the cytoskeleton like some of the other organelles. Also, mitochondria and chloroplasts reproduce independently from the cell. They are not dependent upon the division of the nucleus. So skipping ahead, how did we get this vast diversification of animals then? If we understand how eukaryotes came about, and we understand that it was the photosynthetic prokaryotes, the cyanobacteria, that allowed the eukaryotes to evolve, what about this huge diversification of animals? Within 10 to 20 million years, which is such a short period of time in geologic time, most of the major phyla of animals appeared in the fossil record. What you see here is millions of years ago, the late Precambrian and then the early Paleozoic, or the Cambrian period. This is known as the Cambrian explosion. Look at all of these animals that came about. Sponges, cnidarians, echinoderms, these are your sea star. Chordates. Now chordates, we belong in that group because all of these guys basically have a spinal column or spinal cord. Brachiopods, annelids, these are worms. Mollusks, these are going to be your squid, etc. And arthropods, so many of your insects. And let's take a look at some cool pictures showing you some of the artists' renditions of some of the early life based on fossil evidence. Some not so far-fetched from what we see today. So of course, if we understand that life changed over time and started from inorganic materials, we would question, well, if life could evolve from inorganic material, could it elsewhere? And that's certainly a fair question. Is there life elsewhere? And would it look like life on Earth? Perhaps. I encourage you to read a book called The Cosmos by Carl Sagan, and he does a very elegant job of explaining the vastness of our universe. And just by sheer numbers and by chance, it would sure seem like it would be impossible for there to not be life elsewhere. Sometimes it's hard for us to wrap our minds around that because we feel so special, and we are, but there's nothing wrong with being curious.